You can silence that, I'll keep talking. Great. And also he is very well known here regionally and also throughout the world. He's one of the most collaborative people that I know. He's also been highly prolific over the course of his career and has also been a great mentor. I may be biased though, because he's my mentor and has been the mentor of many people in this room. And going back to that collaborative nature and the sort of impact that he has had, you can see that reflected in his many publications, over 330,000 citations, recent publications in Nature, Science, PNAS, and Cell and related journals. And then also very well funded. So his current funding status reflects sort of the diversity of these interests in systems genetics, and his collaborative nature as well. So he has funding from both the National Cancer Institute, National Institute of Drug Abuse, and also National Institute of Aging. And so today he's going to fill us in on some of his sort of, I can't say recent, it's like an expansion of work that has been going on for some time, looking at the genetics of mortality. But before we do this, I thought it might be interesting to take a pop quiz to see how well you really know Rob. So I wanted to sort of query the audience in terms of whether you think these three statements are accurate. So A, he was once bitten by a venomous cottonmouth while running barefoot in Shelby Farms. And I know we talked a little bit about this with the graduate students. B, he received a BA in psychobiology in 1975 from the same university that I had a BA in, the University of California, Santa Cruz, where the official mascot is the sea lion. Or C, he once challenged a colleague to a push-up contest in the halls of TSRB. And Rob, you can't answer. Okay. So how many people are going with A? Okay, that's good. You're wrong. It was a copperhead.
Didn't see it.
No, but we think maybe it was a juvenile copperhead. But so it's partly true. Okay, how about B? Right. Yes, banana slugs are actually the official mascot, but the rest is true. So then C. Only 2, 3 hands for that? I mean, okay, good. All right, thank you. Yes. So that may be a better like segue into like maybe like the health span, but still we're gonna move now into the genetics of mortality.
Hey, you need to plug a contest.
Okay.
Yes. Thank you. That was a very fun introduction. My success, there has been of it is due to the collaboration, and a lot of what I was credited with. I take advantage of it, I guess. But yeah, I'm going to tell you this story, and I don't know why it's small there. I guess I have to take control here.
Yeah.
Okay, there we go. Okay. So I'm going to tell you, Megan said this has been relatively recent, but also it's a 10-year project. We started this particular study of Het3, Shahida and I, and some others, 10 years ago. And now we're finally ready to publish phase 2. There was a paper in 2022, but this paper is much heftier. For those of you, a little closer? Oh, wow, usually I'm way too loud, but that's good to hear. Yeah, so this paper is much more powerful. The first one was more proof of principle and some cool lip gloss that got it into science. This paper that we're working on now actually delivers the goods. So the question is really, what is the genetic underpinning of mortality? And this is crucial, obviously, for human diseases that are mainly age-related, but it's also crucial for understanding basic biology, evolutionary biology of metazoans. You really need to know what it is that controls mortality rate versus reproductive lifespan. That's a critical parameter in evolutionary biology. And a lot of people have weighed in on this. I just want to thank the first three in particular, Danny Ahrens, who's done almost all of the statistics and a lot of the genetics, David Ashbrook, who until just a month or two ago was here on campus, but now has accepted a position at the University of Edinburgh, and Shuhita Roy right there, who's done all the heavy lifting in terms of keeping track of the data, and there's a lot of data, and keeping track of the tails, and there are a lot of tails, about 25,000 of them, and the DNA sample processing. So, but a lot of other people have contributed, and I'll talk about several of them as I go along. So genetics of mortality. The typical way to present this would be to talk about genetics of lifespan. You will find hundreds, if not thousands, of papers on lifespan as an outcome measure. You will find very few on mortality and almost zero on the genetics of mortality. And the simple reason is you need a ridiculously large sample size to do this. And you can't really even do it with large sample sizes. Come on in, Arthur, find a seat. So sample size has been, one of the students earlier today asked me why we switched from BXTs to these UMHET3, and the answer is sample size. You just can't beat it. So I'm going to start with the conclusions because I tend to wander. The medical students hate me as a lecturer because I won't say things twice usually, and I will go off on a tangent, and they wanted to flay me alive after I taught genetics to them. But I thought I would start with the conclusions like you're supposed to do if you're a good lecturer and come back to that. So bottom line, if you fall asleep because you just had such a good lunch, here's what you need to remember. We've mapped 59 loci that modulate, I shouldn't say control, they modulate mortality rates or lifespan or life expectancy. Take your pick. We don't know what they do yet. So this is highly frustrating to all of you who are more molecular biologists. It's also even frustrating for me as a geneticist, but you'll see that the genetics itself can give you quite a bit of a heads up on what you might be looking for mechanistically. Females and males are just totally different species that get crammed into one genome. And I didn't used to think this was the case for most of the traits that I studied, but for reproductive fitness and mortality, which are sort of wed together, it's really one thing. Reproduction and mortality are tied. Here, you've got two different beasts, males and females, that have to share the same genome to propagate their genetics. And oh my God, it's so different in mice. Now, I'm not saying it's going to be different in all species, but mice have what a male does and a female does in terms of their life history, radically different, much more so than humans. You'll see that. Environment plays a major role, as do gene bioenvironmental interactions. I'm going to say very little about this in the talk. I think I have one slide. It deserves a whole talk. It's going to be more and more important. But my job as a geneticist is to get the genetics done and then explore the G by E interactions, which are going to be huge. And all of you know the heritability of longevity is not great. It depends on the environment. If you're going through the First World War and the Second World War, heritability for longevity is basically a nonsensical concept, just like it is in Ukraine today. I want to get the genetics nailed down as well as we can in mice, where we can control the environment. And then we can add environmental perturbations on top of it, like Shuhita is doing with high-fat diet. Bainey's doing it with a high-fat diet. There are all kinds of things you can do on top of that. Mortality is modulated by many interactions, gene by sex, gene by gene. I'll go through that quickly. And then I want to end on an upbeat note for Andrea, who is a biohacker who's exploring options of extending his healthy lifespan, as am I. So I'm my own guinea pig, and talk a little bit about rapamycin. And so those are the conclusions. And here's the outline of the talk. Genetic question: what DNA variants modulate mortality rates and lifespan. So that'll be the A set of section. That's the longest part of the talk. So that's about 12 slides. Life history questions. What are the differences in causes of mortality between sexes? That's what I just alluded to, that you've got two species crammed into one. Metabolic questions. What are the negative? Why is there a negative relationship between body size and lifespan? So it turns out this is pretty forceful in most species early in life. So those individuals within a species that tend to be smaller tend to live longer. You probably know this from dogs. Small dogs live longer than large dogs. It's also true in humans. It's true in many metazoans, not all by any means. And of course, some species don't have determinate growth. So it again doesn't make sense in those species. But we'll look at the genetics of that, because it turns out that there are a separate set of 30 loci that control that relationship between size and mortality rates. What is the genetic architecture of Vita and Soma loci? So I'm going to tell you about these two types of loci. Genetic architecture, and I put it in scare quotes because I want you to be scared about it, because genetic architecture is static. So a building is a relatively static entity. Genetic architecture is almost a misnomer, because genetics is, if anything, the ultimate fungible kind of choreographed entity. And as an environment changes, the architecture changes. So but what I mean by genetic architecture here is mainly interaction effects. So higher order interactions. The evolutionary question: what are the evolutionary causes of aging? And I'll just touch on that and show you just a few slides. And then, can we extend lifespan and resilience with interventions? And again, one of the students asked, well, you don't just want to extend lifespan, you want to extend health span, healthy life. And that's I share that desire myself, obviously. First, in order to understand what I'm going to tell you about this particular genetic study, you need to understand the cross. Like every one of you, the mice here that we're going to study, which are called these UM3 or second generation, they all have 4 distinct grandparents. Now, you might say, duh, everybody has 4 distinct grandparents, but that's not true of mice. Many mice have the same grandparent that they are, or if they're an F1, they have 2 shared. So what we start with is a maternal grandmother, Babsy Bailey J, a maternal grandfather, Black 6, and then we have a paternal grandmother, C3H, HEJ, and a paternal grandfather, DBA2J. So that's the setup, and we make one set of F1s and another set of F1s, and then we throw those into a cage and make a bunch of babies. And these females here, those are the dams of the cross. They're incredibly prolific. Each litter is about 10, and they'll produce 10 litters of 10, and they'll do that in 365 days. So this is going to be relevant when I talk about sex differences. Female mice of this type are like just beautifully constructed reproduction machines, 100 babies in 1 year, and they take good care of them. Of course, the male mice don't do anything to speak of. Not too much unlike humans, I might add. So anyway, you end up with these progeny, and you can see that they're segregating for white chromosomes, black chromosomes, whatever color that is, and beige chromosomes. And so at any one locus on a chromosome pair, you can say it's brown-black, it's brown-white, it's black-black, et cetera, et cetera. And each one of these distinct colors, I refer to it as a haplotype. So there are 4 haplotypes, and then there are 4 genotypes also. And that's just a link to the 2022 paper. This is what we have, and this is Shuhita is the maestro of this. We have 24,000 tails plus another 1,000 ear snips, so about 25,000 samples with longevity data. the resolution of the het3 for genetics is really, really lousy compared to, say, a GWAS. But that's just the way it is. But while the resolution is lousy, the power is enormous. So in human genetics, nobody even dares to say the word epistasis. Nobody, almost. Whereas in this study, we have over 300 epistatic interactions. So the power is there, the precision is not. And good things are going to come. So collaboration with the ITP, we did not generate these mice. We did not pay for their per diems. We just paid to genotype their tails. So it was just a wonderful collaboration. We don't know, so I can expect many of you will ask me questions about the het3 and why they die and how they die. Nobody knows anything about this. It's just an incredible blank spot in this research program. So here's the lifespan of these mice. They live to about 800 to 900 days. Males slightly shorter than the females. Here you can see a standard Kaplan-Meier curve where the females diverge very early at ages of 200 to 500 days of age. You can see the females keep chugging along, the pink line at the top. The males are busy killing each other in their cages. They're housed three per cage for the males, 4 per cage for the females. No wounding, however, so the males are not literally killing each other. It's not like they're blood marks and you need a detective to figure out who killed who. It's a subtle, probably a stress related, but this is me speculating, as Megan pointed out earlier. Then they converge, and actually at the tail end here, there's essentially no difference in male and female life expectancy at old ages. And you can see that here pretty well where this is that same kind of graph, but now you're looking at the number of animals that went into the study, 6438 at puberty, P42. And we ended the study when only 559 mice were left. So now in a typical study, I wouldn't even show you that. I would just say, hey, we have 6,438 animals for which we have lifespan. End of story, we're gonna map it. No, we're not doing that. We're taking every one of these survivorships, all mice that lived to 42 days. There were a lot of mice that died before 42 days, maybe in situ, maybe in the uterus. We have no idea. There could have been another 20,000 mice here. But we're going to start with these and say, of this survivorship that made it to puberty, what does their map look like? Of the animals that got to 365 days, which is most of them, what does their map look like? And that's what their map looks like. So we truncate at 365 days. We say, we're going to throw all of you guys away and just consider this one horizontal line at 365, and we're going to map that. And then we're going to map 15 days later, 15 days later. There are a total of 72 strata that we map. So that's what we refer to as an actuarial mapping strategy. You can just think of it as progressively older and older survivorships. We're going to ask, how does the map change? And you can see from this that the map changes dramatically for some loci, not all of them. But this is a locus we'll talk about, Vita 1A. It's pretty consistent. You see it at early ages. You see it all the way to 900 days of age. Here's a locus on chromosome 2. You don't see it at all at 365 days. But then up at 700 days, it's a whopper. And here's one that you see, again, pretty early, but you don't see it late. Some of them you do see late. This doesn't show late only loci, but you get all kinds of flavors here. And I will take you through some of these. Now, yeah.
So is it that they're acquiring those, or is it you're selecting for the ones that had them?
Are we selecting? Mother Nature is selecting in the sense that some mice die earlier. Is that what you?
So just so
Okay, one second. Let me go back a slide. Okay.
the large cohort, you're not able to see that.
Oh, yes. Okay, you're asking about this not being there. It's because when you add in the few mice that die in that early era, and they're all male. It basically wipes out the locust. And then when those males have kind of gotten it out of their system, these are the Vikings that went a Viking and got killed entering Scotland or something. Then you see a different pattern. So it's more like an exposure, archaeology. Now I showed you this fancy. Say that again. In that case, very small. Yeah, but when you get to 900 days, they're dying big time, and then it really does change. What we would like to do, we haven't done this very as well as we'd like to. Instead of talking about actuarial survivorships, all mice, all humans that live more than 50, we would like to actually get an instantaneous mortality estimate, but that will take even larger sample sizes. So we started doing it, and this just gives you a feel, and the whole paper has a lot on this. So this is male mortality for one specific locus that I just showed you in the previous slide. But if you hadn't memorized it, you're forgiven. This is the Vita 4A locus. You can see it's pretty strong. It's actually pretty strong at both sexes. And what you can see now, we're going to do it instead of truncation age. This is just regular old age in days. And you can see that if you inherit one haplotype, the D haplotype, you're more likely to die. So this is increased mortality, and that's also increased mortality. And this is sort of baseline mortality. More mortality for the D, then later more mortality for the H. For the C haplotype, you can see there's a lot of mortality at about 700 days. So that's what we'd like to understand, is how the mortality fluctuates across age. And to do that properly, 25,000 mice will be a real bonus compared to what we are doing now. The reason you can see that this isn't adequate, these are the actual points, and this is a smoother that we fit through it. And the smoother looks okay. I can tell a story with it, but the reality is it's messy data still. Conventional QTL map. Now I'm getting you ready for the sex differences. So this is the locus that we did was misdefined. This is the locus that was basically didn't exist at 365 days, the green locus. So combined, there's nothing there. There's a big female locus there. There's actually a pretty big male locus. You put them together, the male and the females, there's a genetic interaction there. If you throw it together, it wipes it out unless you fit the interaction term. This does not fit the interaction term. I'll show you what the interaction term looks like in about 10 slides. But just remember that if you're not fitting interaction terms for sex, you can get in real trouble if the males and the females are fighting in some biological context, here in a genetic context. Okay, so you remember the actuarial map? Well, maybe you don't. Let me go through one of these actuarial maps. So you are going to issue insurance policies to mice at 42 days of age, and you happen to have their genotypes. This is illegal, by the way, to do this for humans right now, which is a good thing. But so at 42 days, they hit puberty. So it's a 13-year-old human. You genotype them and you say, oh, you have the BD genotype. I'm sorry, your lifespan is going to be 30 days lower than everybody else, so we're going to charge you an extra $300. On the other hand, if you had the CH genotype, you're above average, so we'll give you a break of $2. That's how an insurance agency would work, right? So what you're looking at is the expectation of longevity at this age. And you can see that this particular locus, Vita 1A, has a fairly consistent actuarial profile. Well, there's a glitch here, right? So the BD locus or BD genotype catches up. You know how it catches up? Because a lot of BD animals die. And so the survivors now say, no problem, I escaped my childhood lethality phase. So this upward means they died and now it's sort of stable. Now they're starting to die again and they're catching up. So paradoxically, if things look like they're getting better, they're getting better because you're weeding out a bunch of mice. And so this is about as stable an actuarial map as you can think of. But what do you think the mortality rate looks like for these? So you expect to see a big bump in mortality rate here for BD. You expect to see all kinds of mortality stuff happening here because the rate of change is what matters. I'll skip that for efficiency. That's just the old school way of presenting these. So here, just to remind you of the haplotypes, and here is the same locus, Vita 1A, and now we're actually looking at the, I would call it instantaneous mortality, but it's anything but instantaneous. Basically, it's averaging, smoothing over 60-day intervals. And then so each point it's shown as a point, but it's actually a smoother. And you can see that the D haplotype does bad things early, it does bad things here. The H haplotype does bad things here. And we've got the females up there and the males down here. So now you can kind of look at this and compare the mortality of females on top, males on the bottom, and you can see a lot of male death going on here, nothing for the females. So this is a locus you'd say, okay, there's a sex difference there in early male mortality versus female non-mortality. Whereas both males and females, the H is not a good allele to have after 1,000 days. It's going to kill you at a higher rate than other alleles. So that's a kind of a mortality rate. And let me just brag a little bit about this. This has never been done, even in Drosophila, even in C. elegans, even in yeast. So not sure why. I think people are just thinking about lifespan as this reified entity that you had to study. And nobody in the last 20 years has really thought seriously about mapping mortality. And so even Danny and I didn't have a clue what we were doing when we started doing this. We thought we were mapping lifespan. Then we added the actuarial. And we said, no, we're actually mapping mortality. The types of Vita loci can be categorized. You already saw Vita 1A. That's Vita 1B over there in the upper left. Very much the same. If you were issuing insurance policies to mice at 42 days, you wouldn't have to change your rate until about 700 days, because pretty much the same. But then after 900 days, you go, whoa, I got to reissue you your life insurance policy because things got very different. There are some loci, the ones that kind of the best loci, the ones that we really hope to find will be loci that control the rate of aging. Why is it that a mouse lives so long, maybe 900 days, but a bat that's even smaller lives 10 years? That's a rate of aging. And we'd love to know what's controlling that within mice. We don't know, but they would have a relatively constant slope on these mortality graphs. They'd basically be this way or that way and tell you that that locus has a relatively constant effect. Then we have loci that are mostly male specific. This Vita 1C locus, look how it wipes out males only up to 600 days. And if you inherit the BD genotype again, woe be unto you, because it's going to cost you 35 days relative, well, it's going to cost you more than that relative to BH. So they're very strong effects. I would love to know the mechanism. My suspicion is that this will be stress hormone systems related. But then after 600 days, everything is chill. It's like, okay, vacation time. I'm just going to sit in my villa and drink pina coladas until I croak. And so all of them basically do the same thing. This is a very interesting, and by the way, the reason I'm telling you this is not just to classify them. This is really important for evolutionary theories of aging, because evolutionary theories of aging make all these sweeping claims about there should be gene variants of this type, the other type, the other type, without any empirical evidence. It's quite remarkable. Here's a type of locus that only acts after about 1,000 days. Here, Eric. Adding to the cacophony. So these are the real late acting loci. And, you know, Mother Nature doesn't know anything about these loci, right? They killed mice at 900 days. What does Mother Nature care about a locust that kills a mouse at 900 days? It doesn't. It's basically completely invisible to natural selection. This is a weirdo that Danny and I must say, we puzzled over this for several months. A locust that has a biphasic mortality function. It turns out it has an easy explanation. There's just two mortalities that are slightly phase shifted by about 100 days. And it gives this weird effect when you map it at the genotype level. If you look at the haplotypes, it makes a hell of a lot more sense. So this is actually that locus and in males. And you can see that you have funny things going on with H. It's hard to tell, but there's enough of a phase shift here between high mortality in the C haplotype and high mortality in the H haplotype to generate that kind of biphasic relationship.
It's showing a lot of these guys, just like I want to make sure fully capture. So to mortality, you got like a increase in mortality, even if you go on the top and
but-
the bottom.
In both of these, there's an increase. So it's a messed up scale.
Oh, it's okay, and then the effect size is clearly different, that's something which is beautiful.
Yeah.
Just like it's, however, there are trends of mortality by times, right?
Yes.
Dependent of the year, is that correct? Yeah, you still see spikes, and then the intensity of the spikes, it's like for the C allele, it's almost plus 0.8, and for the H, it's 0.4, it's half.
Yeah.
But still, you get the spike at the same time, and this has been consistent.
Oh, yes.
Right, so what's the explanation?
yeah.
It's like, you know, they have waves of time, so is that correct?
Yeah. If you're puzzled by this and you have a good question, I still have a hard time visualizing this. I think we need mathematical models to do this. One of the struggles here in reconciling these two is that this is using genotypes and this is using haplotypes. So you have to kind of integrate the effects of both of those. We'll come back to it and we can chat, see if you can find a better method to do it.
Sorry, what are we seeing on the y-axis here? Is it days or?
On the y-axis here.
That's exactly why.
Oh, thank you, Ramesh. I need more nicotinamide. Okay. In this case, it's a genotype effect in days. Over here, it's mortality difference, mortality rate difference. So delta percent mortality. Yeah, now if only you'd said the abscissa or the ordinate, I can never keep those straight. But, okay.
What's the resolution of the haplotype?
What is
What's
the,
resolution of the haplotype? How big of the haplotype?
oh, the resolution of the actuarial mapping is 15 days.
Haplotype.
That, okay. That I can't discuss because it's embarrassingly large. It's essentially an F2. So haplotype blocks are going to be half a chromosome. And the only way we get away with this is because we've got 6,438 animals, which is not nearly enough. Yeah, great point. Okay, memorize this, put it in your eidetic memory. If you can't see it well enough, then put this in your eidetic memory. I just want you to give you a feeling, and this answers Xusheng's question, very embarrassing question, by the way, which is how big are these? So here's the size in megabases. So this is Xusheng's answer. Should have waited one slide, Xusheng. So the average of all of those is about 35 megabases. Totally embarrassing. But the very first one's 8 megabases and only has 14 protein coding genes in it. We have 2 really good candidates that we're thinking about knocking out. But what you can see, the lot scores are pretty awesome. So the lowest we let in is about 4. We have to correct for the actuarial method because the actual method is testing again and again and again. So there's a multiple test issue there. And we use a Cauchy correction that Shonak Sen developed for us because I didn't know how to do this. But it basically asks how much dynamics do you have in any one locus? And if the dynamics are just flat, there's no correction. If it's going up and down, you have a more intense correction. But these are all also significant with the correction. I just want to focus again on what Xusheng mentioned, the large size. But here's another thing. When do they start acting? So you can see most of them here, by coincidence, act starting at puberty, but one of them starts way after reproduction. 575 days
There's
is,
some threshold on when we see a positive angle delta of the life expectancy.
is there a,
Is it like, how do you determine 42 days?
okay.
You-
What we do is
it's a
we-
range, right?
Yeah. We basically look at the LOD score. So we have, in the same way that you do a confidence interval for QTL, you'd say, okay, I've got my peak. I'm going to take 1.5 LOD drop on either side. We do a variant of the same in the chronological dimension. Yeah. And sometimes we have to have a fudge factor in there because sometimes things get a little awkward. This is all I'm going to say about non-genetic factors because I'm still on A and it happens to be getting close. I will say that look at so the green line up there is the green boxes are the environmental variants, VE, variance environmental, and the red is variance genetics. And you can see that the environmental variance for the males is quite high, roughly 42% at early stages. Look at the variance for the females, only 30%. And I think that is because the main environmental variant is the other males in the cage. It's not literally, when lights went on or off. The female's much closer of sources. This is epistatic interaction, which I'll show you later in the talk. But that's genetics. So what you cannot see very well here are these very faint orange, which is unexplained variance. And then here's another source of explained variance. So now we're going to take the environmental variance, these green bars, and take it apart into the 3 factors we know about. Half of these animals were absolute controls. Nobody touched them with anything. The other half of the animals received a drug treatment that was ineffective. And so we just took the ineffective and said, okay, we'll correct for the fact that they got an ineffective treatment. But you can see that the drug effect is sizable, 14% of the variance is accounted for, and even more sizable in the females, 14-28. So twofold difference there in the drug effects between males and females. Oddly enough, drugs, any intervention has almost any intervention has beneficial effects on male mice. Very few have a beneficial effect on female mice. Nobody knows why. It's like an incredible mystery because there are 15 drugs now that have strong beneficial effects, of which 14 are male, much stronger in males. Rapamycin is the one wonderful exception. And then cohort year. So this study was done over roughly 10 years. Cohort year doesn't matter. But environment is important. And G by E, of course, is going to be important.
I doubt you have this information, but in terms of how they manage the cages, like as the males die, obviously you have fewer mice. in the
Yes.
cage.
Yeah, great point.
Often for cost savings, you have females who find them.
Yes, we do that. Yeah.
You do that. So
You can't, they don't. So they have very rigid, so it's a great question because it's a confounder and it's a confounder there's no solution to. You can either try to buffer the difference in cage density or you can just let them live in their cage. My suspicion is the reason the males do well late in life is because they're going, oh my God, thank God Cain finally died. Whereas the females would like to have their sisters around. So yeah, but the way they designed it's very rigid. We do what you said. We just throw mice randomly, which is maybe a problem, but
Changes in cage numbers, what's time? Is there any way you can?
They record who's in every cage every day. So if a mouse dies, so we can begin to ask questions about indirect genetic effects. We haven't yet because we only got the data from one of the 3 sites.
The cage can be a control variable. So every cage day, it's like control.
Yes. Yeah, I'll have to think about that, Eric. Yeah, yeah. Okay, so what about mechanism? It looks like I'm going to get through the A section in my talk. I'll just skip through this, just promising that we're working hard on it. We've ordered some conditional knockouts with Aman and Shahida. We have our favorites. We've been testing it by doing C. elegans motility assays. And we've also been testing it by doing Mendelian randomization using UK Biobank and have some good candidates from UK Biobank. That you can do at scale. Sex differences. Okay, so let's scale at least through sex differences. I mentioned how this is huge. I mentioned this effect where not accounting for the interaction term kills the locus when you just use sex as a cofactor. You have to use sex as an interaction term, not as a cofactor. This is what the maps for the females and the males look like. And I realize that the resolution isn't there to really resolve it, but here's a male specific chunk. You can see that the females have much more going on here. Males don't have much going on here. Here we have some strong male activity on 4, nothing for the females. Frankly, it's shocking how little overlap there is. There is overlap. So of those 29 loci, about 7 have really good overlap, Vita 1A, Vita 1B. But I think the take-home message is how different this is. And we're talking about something quite fundamental, mortality. You'd think there'd be a common denominator there, but there isn't. This is the formal test for the sex interaction term only. So we're only showing you the map of the interaction. And this is the chromosome 2. It's basically this more than a half of chromosome 2 where there's just a ridiculously strong sex interaction effect that you can again see here. So purple is the interaction term. Red is the additive effect of the females. Blue is the additive effect of the males. If you add the males and the females without the interaction, you get that. So they're basically polar opposites, and I'll show you that in the next slide. So here they are, the opposites. Have a look at the females over there in the upper left, BH and CH at the top. Whoop, over here males, BH and CH at the bottom. So they are literally flipped in terms of their effects on mortality at that age. And here's just a kind of standard gene by sex interaction plot where you can see things are wonky in terms of all these crossovers, males in blue, females red, and the different genotypes. We have 4 different genotypes, and you can just see how it flips back and forth in a way that says, I'm interacting. Here's another way to look at it, at the mortality rate difference. If you're a female, D is bad early in life. If you're a male, H is bad, and then again flips. Most of the action here is D versus H. You can see there's not much going on at the top. So it just shows you, if you needed an example to give to any students of why you have to fit interaction terms, that's it. Okay, we did it. Now, part C, what are the soma loci? I should make him wait. What about the Y?
Why?
So we know nothing about Y. We know nothing about mitochondria because they did the UMHET3 cross in one rigid 4-way pattern. Of course, they're 4 times 4 minus 4 divided. Well, yeah, so there are 12 possible ways to do the UMHET3, and they only did one. And Shahid and I have tried. and Bainey have tried some of the others just to see what Y in mitochondria might do. There's a kind of inevitable confound between.
Because it's from set, it takes differences and then the Y is biased to one species
Yeah.
might have this kind of maladaptive.
Yeah.
Just
Yeah, yeah. I think that this would be a real concern if we'd use crazy mice. There are lots of crazy mice that people used in their research. Wild caught mice, or North African mouse spreadus. These are kind of, run-of-the-mill mice, still segregating for 11 million variants. Just like humans are basically, all of us are run-of-the-mill humans. We're very similar. These mice have more genetic diversity than all of humanity. 11 million common variants segregating with maps of exactly 0.25. So it's a huge amount of genetic variation. So third story before Eric asks me another question. What are Soma loci? So we got 30 of these, and they control the correlation between body weight, which is the only measure we have. Body size might be better, but we have body weight and how long did a mouse live? Body weight at this age, that age, the other age. So here at the top is body weight at puberty, and the correlation on the y-axis, your ordinate, is -1 roughly for males and females. So weak. But then as they get older, as they get to about 900, 1,000 days, basically it approaches 0. So there's no correlation. But look at the 365-day weight. So this is kind of at the tail end of reproductive fitness for a female or a male mouse. And the correlation there is gets down to 0.25, 0.3 at that age. I'm sorry, I screwed up the scale. 1, 2. Yeah, for the males, it gets down to 0.25. That's a pretty strong correlation. Doesn't look like much, but I'll show you genetically, it's actually quite forceful. And this is for all, this is not for a locus. This is body weight versus life expectancy. So what we're doing is taking these correlations and asking, can we convert them into genetic loci that actually explain this factor? And you can see that for those of us who are older, it's very encouraging to know as we get older, it's actually beneficial to have to be heavier, which kind of makes sense because if you ever seen your grandma or your grandpa, kind of go off the cliff, they get skinny typically, unless they gracefully die of a heart attack in 2 hours, which is how I want to go. So this is what the Soma loci look like as QTL maps. The top one is at 42 days. The bottom one is 183 days. Obvious question all of you should be asking probably is, are they the same as body weight loci, or are they different? Are they the same as Vita loci, or are they different? They're different. They overlap some of the Vita loci. Of the 30, maybe 7 overlap Vita loci. They don't overlap body weight. So this is something different. And let me just show you what they actually look like, because who other than Rob and a few other hardcore types likes looking at QTL maps? Now there's some data. Oh my God, that is so beautiful. Bainey, don't you agree? Is that the most beautiful table you ever saw? Okay. Okay. All right. Well, here's what they actually look like. So here are the correlations for the 4 genotypes. I don't know where, 4 genotypes. And you could, at 200 days, and then we're asking, are there differences in those correlations? And you can see CH has a very low correlation, less than -0.2, whereas the CD gets close to -0.4. So that's the difference in correlation. And we're asking, is there a locus in the genome that's accounting for that difference between CH and CD? And the answer is yes, there is. And that would be the Soma3b locus on chromosome 3. So now we have a locus that's actually controlling a pleiotropic interaction between body weight and lifespan. And so this is kind of a classic problem. Everybody who's done genetics for more than a couple of years heard about pleiotropy, the fact that one gene can have multiple effects. So this is one locus that has some effect on body weight and some effect on longevity, and we see it as a correlation. And here's the, I'll be with you in a sec. So here's the male, females, nothing going on.
you said there's no plant, but if you, instead of looking at loci individually, you place them within biological pathways, are there biological pathways?
Oh, yes, then for sure.
If you can have
Yeah.
genes, two different points of biological pathway, and show up in one, and then another one, same biological pathway.
Yeah,
shows
yeah,
up in
no,
different-
absolutely. And we're trying to do the gene ontology analysis of the loci and groups of loci. We haven't, they're too big, they're too fat right now to make much headway. But as the loci get smaller, we may be able to figure out whether we can detect the signatures of those genetic pathways. So yeah, that's the crucial part of getting from maps to mechanisms. So it'll be maps, enrichment, perturbation experiments, and onward. Okay, part D, genetic architecture. So let me just, I'm going to skip this charming, absolutely charming table that I know you want to look at for longer, but sorry, I'm not going to show it to you for longer. I'm just going to say that there are lots and lots of epistatic interactions. I mentioned about 300. The males and the females are worlds apart. So here's a correlation of male epistatic Alod scores, and these are high. So everything above 3.8 is significant. This is the male set of significant epistatic interactions. In red are the female significant epistatic interactions. Notice anything about quadrant 2? Nothing there. There's not a single epistatic interaction that's significant that's shared by both sexes. And if you look at these green points here, they actually show that they're fighting. Those, if you take those green points apart and ask what's going on there, they're actually pushing and pulling apart. This is what it looks like if you look at all the epistatic interactions as a bicycle chart. We refuse to call these Circos plots. They're bicycle plots. So males over there, females over there, grab any one of those traces, which gives you an interaction, say, between Soma 16A and Soma 19A. This dark blue, it's males. Or no, it's Soma, sorry. And then look over on the female. You won't see anything in common. Or you'll have to, you will, they're probably 5% are common, but I'd say the story is, wow. Yeah, you can see the overlap here. The overlap is 2, 78 male, 72 female, nothing shared. Well, 2 shared. Now I have a little, okay, so this is gonna hop back and forth because I didn't know what I was doing with PowerPoint. But what that showed is, this is now you're looking at 905 days. Radically different set of epistatic interactions. Again, nothing shared. 3 overlap, I should say 3 overlap, 63 male, 49 female. So I think I'm going to conclude with this slide, and I'll skip through the validation, the kind of mechanistic stuff that interests everybody, but we're just at, baby steps on that part. So this is probably the most interesting kind of core science for everybody. Why do we age? And everybody, the first theory was it because of feces in your gut killed you, believe it or not. So if you look at the 1911 Encyclopedia Britannica, it'll say that senescence is caused by gut microbiomes, although they didn't have that word then. But these days, it's all the telomeres. They got short and raggedy and things fell apart. Oh no, it's free radical damage, definitely what's causing it. It's redox stress in mitochondria. It's got to be it. Nope, not it. It's all DNA repair. Well, there's pretty good evidence for DNA repair, but if that's true, why do none of our loci sit on top of DNA repair genes? We don't, we haven't ruled it out, but so far nobody has found a natural polymorphism in a DNA repair gene, and we know a hell of a lot about these thanks to cancer, that modulates longevity, normal longevity. I mean, you can have a DNA repair gene that kills you when you're 15 or something. That's a different beast. So we need to think about the why question, and the why question basically takes us back into, big crazy ass theory that a molecular biologist is not allowed to ask. You are not allowed to ask why questions, right? No, no. But, this begs for a why question. So there are three big theories about why we age. It actually goes all the way back to Haldane. Haldane in his 41 Modern Genetics textbook. I hope everybody's read Modern Genetics. It's actually a really good book. But he points out that that you can have a mutation that doesn't appear until late in life. Who cares? Mother Nature doesn't care. Huntington, he actually talked about Huntington as being the best example he knew of. And he's right. There's evidence that Huntington males with Huntington's disease, have higher fecundity than males without Huntington's disease. Isn't that weird? And that's because Huntington doesn't really grab strongly until about 45. So that's an example of a mutation that's beyond the scope of natural selection to impact. So the three big theories are, this theory basically came to be called the mutation accumulation theory of aging. I hate the word mutation in here because they're just normal variants. They're not really mutations. They're just normal variants. They may be good or bad early in life, but they have bad effects late in life after reproduction, which I hate to tell everybody this, but you begin to fall apart when you're 21. Does everybody know that? Yep. Yep. Sorry. Genetically speaking, that's the beginning of senescence. I know you can still win the gold medals at the Olympics for the next 10, 15 years, but yeah, I'm just getting even with everybody because I'm a little bit older than 21. Antagonistic pleiotropy really says you have to pay, you can have a beneficial effect like Huntington that I mentioned, but oh my God, it's going to do something really nasty late in life. So that's the antagonistic pleiotropy theory that was formalized by a couple of people, but George Williams is kind of the canonical example of that. That says good effect of this allele early in life, bad effect late. And the Soma loci basically scream that. They say, yes, it's better to be a big male early in life and then bad things happen. And then this disposable Soma theory says, who cares about our bodies? It's only our gametes that really matter. So let's put all of our energy into gamete production. And, if that works, we can just relax and not spend any time maintaining our Soma. And so that's the disposable Soma. Soma theory, they all kind of weave together. They're not really as distinct as stated here. And I just want to point out that there is no one size fits all. We're just studying mice. And oh my God, these other species are so different. So some fun paper, books, great paper by Peter Sudmont and colleagues on this. Rockfish are really cool. And I'm going to skip the drug intervention stuff. Sorry, Andrea, I'll have to tell you later, but start taking your rapamycin immediately, if not sooner. It does good things unless you start taking it when you're a mouse at 900 days, it's not going to do much for you. But if you even take it at 600 days, which is pretty damn old, it'll do good things for you. Okay, so conclusion. So genetics of lifespan is actually the genetics of life history. That's what I've learned over the last year. I've also learned that male and female mice have radically different life histories. This is true of mice, maybe true of some other species, probably true of chimpanzees, probably not of bonobos, which are much more cooperative. I kind of like this metaphor of one genome, two species, two sexes. Sexes are really quite different. We have plenty of loci. We don't have enough precision, but we've got plenty more mice, and we're going to get to mechanism. And then I'm going to be in the pool in 10 years, Shuhita. I'm just going to be in the pool here, rejuvenating. This famous old where the old people get dumped into the pool. It's full of rapamycin and nicotinamide. And they come back and you can't see it, but they're people heading into the woods together. It's disgusting. It's Renaissance pornography. Okay, thanks.
So I really enjoyed that. That was really great. So a question I have is, do you think geneticists maybe are too biased to the nucleus and basically are not looking at other organelles? I mean, you mentioned mitochondria, but maybe aging is the mitochondria, maybe aging is the endoplasmic reticulum and protein folding, maybe because there are a lot of the things are like telomeres, DNA damage, oxidative stress to the DNA, and maybe it's the other organelles that are being ignored. And sort of related to that, is there a way to look at your things that are clustering in terms of two things? One, where they go pathways, or three things, pathways, but also organelles. And also, Ramesh, I think, may have left, but looking at ER and estrogen receptor and androgen receptor responsive genes, things of that nature.
Yeah. No, absolutely. That's the next frontier. What we're trying to do, Kevin, and I think all of you had this question, Ramesh had the question, is when do we get to understand what is happening? I'm not telling you what is happening here. I'm telling you where the source of the variance is at the DNA level. Presumably, of course, DNA variants are kind of upstream instrumental variables, in my opinion. It's almost as good as saying I dumped rapamycin on this mouse. So it's an instrument that I use to get at the raw causality, but I don't know anything about mediators or mechanisms. And that's, I will say, so the way you started your question, you said it has anybody. I would say 99% of geroscience is people going, I had a dream last night that it's telomeres. So I'm going to look at telomeres. Oh, no, I had a dream last night that it is, mTOR signaling and this particular gene. And if you look at last week, there was a paper that looked at Greenland whale sharks and found one gene that miraculously, Kevin, accounts for the fact that they can live to 392 days. And if you believe that, there are several bridges Donald Trump can sell you. So yeah, so I guess I'm hesitant to talk about biomarkers and mediators at this point until somebody has nailed down the natural variants that cause aging, whatever they turn out to be. I'll get you.
I think looking at epistasis, what's the general direction of their interaction? So are they additive, or different loci? Are they additive? How additive on average they are? Are there destructive interferences, diminishing returns? Are there any clusters in their interactions?
Tricky question, Michael. Probably most people didn't get that, but there are many types of epistasis. We don't have access to other types. It's a very simple cross, so we only have additive epistasis. We can't even look at dominance type epistasis. Well, all of the variants that I showed you in that heritability plot, that's all additive epistatic variants, only attributable to Vita loci. So we took 29 Vita loci, said, what's the additive variants that they throw in? But you're right. If you were to do a messy cross, just throw all of those 4 genomes and just let them go for 50 generations, you get access to all forms of epistasis, and you get access to Eric's question about Y chromosome, mitochondria, much besides. One second, Hao Chen hasn't asked.
So I follow up on the epistasis question. If you can narrow down the haplotype size, do you think you have more epistasis or less epistasis?
I think more. So I think as we narrow down the haplotype block size by increasing the sample size, I expect we will at least double the number of loci we have. They'll basically split apart. So instead of Vita 1A, we might have or Vita 1A1, Vita 1A2, Vita 1A3. And in that case, I would hope if we had the resolution across the board, we should now end up with epistasis that's discrete to Vita 1A1, and we don't see it in Vita 1AB, 1B. Did I get your question right?
Yeah, because I was also thinking that it might be that some of these epistasis are because the haplotype is big.
Oh, that's definitely even a factor for the additive effects. So there are a lot of additive effects that we pick up just because of the luck of the draw, that there are 3 aces under that locus that are in phase. If they were out of phase, obviously it would kill us. But yeah, I think we won't know until we do this better. But yeah.
So clearly Tor signaling is something that you're interested in. Everybody's interested in, looks at longevity. So one of the driving forces of aging that there's a lot of experimental evidence for is epigenome deterioration.
Yeah.
So you get degradation of the epigenome as a driving force for the aging process. And there's evidence from mice to mammals that that is a well, in many organisms, there's a lot of evidence that you dysregulate the epigenome and you accelerate or you reduce longevity.
That's true. So, so I guess so we're going to go around and around. So there's a difference between a really good molecular biologist and cell biologist and so geneticist. And then I just am interested in the genetic causality first. Then I want to get to the genetic mediation effects, which
Okay.
what you're talking about. I would love to. So, for example, let me pick mTOR because we had data that didn't show it. mTOR has a strong interaction with Vita 1A and Vita 1B. It has no meaning. So kind of like that sex effect. If you just map the rapamycin cases, and we've done 1,000, you get 0 signal on proximal chromosome 1. If you look at the interaction, it's a whopper. So there's some interaction there that somebody like you might be able to make sense of.
Well, so what I was going to say that, there's evidence that deterioration of the epigenome is, let's say, a contributing factor to longevity effects. There's also accumulating evidence that as organisms age, there's a drive to increase Tor signaling, especially in somatic tissues. But so there's this general increase in Tor signaling. So we actually have evidence that degradation of the epigenome is a driver of increased Tor signaling. So I'm
That thing for the transcript.
well, yeah, generally speaking, it is. But I'm just wondering this connection between epigenome deterioration and Tor signaling. If you look at these loci, I know that you don't have them down narrowed like at real high resolution, but when I was asking about biological pathways before, you could have genes that are involved in a biological pathway that act in different ways depending on what that gene product does in that biological pathway
At that age.
at that age.
In
So,
that sex.
right. So I'm wondering if you can actually start taking these loci and instead of thinking about them as discrete genes, think about them as biological pathways and then look at their interactions with the Tor pathway perhaps as a whole or even like epigenome specific pathways. Is there a way to do that where you're at right now?
We're trying. So what we've done in the last month is we tried to do a real analysis, pathway analysis. And because I didn't tell you this, but I'm going to fess up because you think we'll on now. So collectively, all Vita loci cover 1/3 of the genome. All Soma loci cover 42% of the genome. So doing a Go analysis, it's like, okay, how is that going to work? So what we decided last week is, well, we'll just look at, is there an enrichment in a locus. If the locus has 200 genes in it, maybe, and MHC, anyone? You know, you look at MHC, it's Vita 17A, duh, it's MHC, it's not one locus, it's going to be dozens of loci, all the way from the T locus all the way to MHC. Similarly, though, the X chromosome in males is a whopping big effect. Hey, that's not a locus, that's all of X chromosome in males because it's hemizygous. So it's disadvantageous to be hemizygous. So, but what we're doing is now looking at the larger loci, asking, do we see an enrichment? Obviously in MHC, we see immune enrichment. But I think your question would be, could you see something that was involved in transcriptional control or, yeah. Or, yeah, so we have done that. If you look at all the soma loci and do that analysis, they're more enriched in metabolism than the vital loci, which you kind of expect.
So is there evidence that the age, so those genes that you have at extreme age, would those be growth promoting genes that may help with frailty, age-associated frailty?
They're not selected, so they're doing whatever the hell they want. They're not going to be adaptive. It'll be chance benefit rather than adaptive. You
Selecting for things that prevent frailty at that age that
Yes, you know, by definition, one of the allele states is beneficial compared to the other. And so if you knew how to pick and choose those late acting beneficial alleles, you could stretch healthy aging, maybe. Shishang?
Yeah, just a quick question. So, Rob, you mentioned 3 series. So, is there any particular reason you didn't mention, you know, chromosome terminus shrinkage and mitochondrial dysfunction?
Oh, no, sorry, I should have, since I have a paper on it or 2 on that. I was just trying to make fun of other people, Shishang, instead of myself. I would hate to make fun of myself.
Okay, yeah. So the fact that you see such huge sex differences, so let's ignore my discomfort over the potential distortion of your cross, right? But whatever, that's unavoidable. So stay freaked out. But I'm gonna assume that doesn't matter. The fact that the sex differences are one of the most notable features of the data you have, doesn't it say something about reproduction and the reproduction of the species of the community is an essential part of aging, or essential that somehow really yeah. So but that was not one of the theories you put forward. Actually, one of the 3 theories you gave, that wasn't really one of them. Which one is it? Yeah. Well, I mean, I know it's out there. I'm just saying that it's interesting that wasn't one you selected to bring forward.
Because there's not enough recognition. There's not enough recognition that mortality is basically a product of sex, which is a product of, well, males and females deciding when to have babies. That's about as simple as it gets. We're just cruising, baby. See me cruise.
So I was going to say, community level, in terms of group selection, that makes sense.
Yes.
Because you can transmit it to culture, but in strict sense of definitely there's no justification to survive after reproduction, not
Right.
for a short time until you're right.
Yeah, but probably everybody's heard about, so what Enza said is right, but I'm sure everybody's heard about the grandmother effect. Supposedly your grandma increases your fitness. The evidence there is really weak. Yeah. Yeah.
Quick. So this may be more of a philosophical and unanswerable question, but do you think you're seeing this because, mice are evolutionarily much older than us, as you point out, because they're very genetically diverse? Do you think it just because the male-female dynamic is basically happens because pretty much they're in direct competition, so never can evolutionary resolve in one direction or the other? Meaning anything that was beneficial to both would be selected over time, but anything that's beneficial to one, but and the opposites.
That's not advantageous because you cannot hurt two.
Right. So but I guess that's my point. Because they're in, if you have a loci that's in direct competition based on sex, it never will resolve in one direction.
Less opposed polarity. So it's a great question. It's not philosophical, but and people are approaching this by rearing diverse mice in wild cages. Wild pens are like an acre in size, and seeding them with like 6 or 8 nodes where mice can reproduce. And of course, the males, big males, get one of these houses and they accumulate like 5 or 10 females, and they defend it to the bitter death against other males who are in others. And then there's some straggler males that don't cut it. So you could begin to approach these kind of population genetics, eco-devo questions, but not with NSF funding going.
Last question. Because Rob is here this afternoon.
Speaking on like sexual dimorphism, I haven't checked it in mice specifically, but in humans, the mortality structure is more basically the same between sexes, right? Most people die to cancers, mostly the same cancers, men and women, cardiovascular, mostly the same. And how do we reconcile it with the fact that the predictors of longevity are wildly different, even though what it manifests as in age-related disease is basically the same?
We don't resolve is probably the quick answer. What's amazing is that mice living in a vivarium and humans living in a Western society like ours, excepting Russia and Ukraine, as you know better than all of us, are, we're in a luxurious position of not dying of predation. Yes, we do die of infectious diseases, but it's relatively mild compared to our ancestors 200, 300 years ago. I wish somebody would do an impossible study of longevity trends over the next 1,000,000 or 2,000,000 years between bonobos and chimpanzees. They live very close. They have very different ecotypes. So south of the Congo is tall forest, and the bonobos live in the forest basically, whereas the chimps are more ground dwelling. The males can't even climb trees really. But the life history of these species, and this is really interesting in terms of pangenomics, they're basically the same species. You can get them to interbreed if you try hard enough. But oh my God, the life histories are completely different. So you'd expect that the genetics would be radically different in 1,000,000 or 2,000,000 years if they keep it up and we don't kill them all, which don't worry about it. We'll kill them all. So it's not an issue, except the zoo animals.
Well, thanks everybody.